This is a presentation of the paper HOX10 and HOX11 genes are required to globally pattern the mammalian skeleton by Wellik and Capecchi in 2003 in the journal Science. And the first thing that's useful to know about this paper is what is the problem they were trying to solve? What was the scientific or intellectual need or basis for this work? And this is taken directly from the introduction of their paper. They say it best that over time, there have been lots of studies where scientists have tried to mutate or remove one or maybe a few different paralogous HOX genes, but they show only relatively minor changes in skeletal phenotypes. So the question then is, how do you get around this issue of redundancy? How do you learn what these HOX alleles or paralogs might be doing if you can't understand the roles of one at a time by getting rid of one allele at a time, or even something a little bit more major like that, like maybe 2 or 3 alleles at a time. So this redundancy between the paralogous groups, that is there a difference between HOX10 and HOX11 paralogs in development, has been difficult to address because you can't get rid of, or at least they weren't able to until this point in time, get rid of whole sets of paralogs at once. So the big question is there a HOX code in development? Is there a particular set of HOX genes that dictates in the axial development from anterior to posterior? Here we're looking at a spinal cord. Is there a code where the expression of particular HOX genes dictates the region of the body that is going to develop? And again, we raise this idea of the difficulty being the presence of paralogous HOX genes. If you got rid of HOX10 genes, for example, is it possible that the HOX11 genes would be able to substitute for the missing HOX10 genes? So to reiterate, there are in mice 4 different paralog groups. There was ancestrally one set of HOX genes, and now in mice on 4 different chromosomes, there are paralogs group A, B, C, and D. And this is the redundancy they're talking about. So for example, for HOX9, there are 4 different alleles of that paralog. Sorry, there are 4 different paralogs with 2 alleles each potentially, because all of these animals are diploid. So that's a lot of copies of the same gene, or what we might assume is the same gene. That again is the question of this paper, is what do these genes and alleles and paralogs do? So that's the experimental objective. The scientists want to know, in this case particularly, how do the HOX10 and HOX11 paralogs influence axial development? And really, a bigger question is, do they influence axial development? There are studies from other systems that suggest they are, and from mice as well, not just other systems. But that's a big question. What do these alleles and paralogs do? An obstacle up until this point, which is why this paper made such a splash when it was published in the Evo-Devo world and the developmental biology world, is that to this point, no one had been able to get rid of multiple alleles all at once in a mouse. You might expect, and it's true, that these are essential genes. If you don't have all of the genes, you can't develop. The mice embryos won't come to term, they won't be born. They certainly aren't developing the skeleton properly, so if they are born and they did survive, they probably wouldn't be able to eat, or certainly not reproduce and make yet more mutants. So the obstacle was a technological one. And in this case, what the experimental approach was there are regions that contain, for example, let's say it's HOX10A. So what they do is insert in the gene a selectable marker. That is a new gene that's easy to tell whether a mouse has it or not. And in this case, if I remember correctly, it was an antibiotic resistance gene. So what you would do in principle is you embed this selectable marker in a mouse, you find out where in the genome that selectable marker integrated, inserted the gene into an existing gene, in this case the HOX10A for example. And that insertion of a gene in the middle of the HOX10 gene, you would predict would prevent the expression, the proper expression, either transcription or translation of the appropriate protein from this gene. So they were knocking in selectable markers or inserting selectable markers into these genes to disrupt them. And that's how they generated the mutant mice that don't produce the proteins that are these transcription factors, the HOX transcription factors. So the other thing that I want to mention before we start looking at these figures is that the authors say in their methods section, they collected between 2 to 7 skeletons per genotype, essentially. So what we're looking at here are embryonic skeletons. So these are not mice that were alive. They were alive, but they were euthanized and extracted as embryos. And then they used alizarin red, which is a dye that stains ossified or calcified bone. And they used, I guess I should put red there as well. And then Alcian blue, which stains cartilage. So they extract the embryos, they remove the flesh, they stain the bone and cartilage, and that's how we can see what the axial pattern looks like of these skeletons. And again, axial pattern is, in this case, relates to this idea of an axial formula, which is the number of different types of vertebrae that, in this case, a mouse has. So in this case, there are 7 cervical in a wild type mouse, and 13 thoracic, and 6 lumbar, and 4 sacral, and then numerous caudal. And so we won't worry too much about those. So what we're looking at here then is 2 different mice, one the control mouse, a wild type mouse with no mutations whatsoever, and a triple mutant mouse, a mouse that has, in this case, lowercase alleles indicate the mutant alleles. So 2 mutant alleles, both mutant alleles of HOX10A and C and D. So they refer to this as a triple mutant. And that's because they've hit all 3 paralogous groups, the A, the C, and the D. But really it's also a, I don't know, what would you call it, a sextuple mutant for 6, because they've mutated both alleles of each of those 3 paralogous groups. So the basis for the analysis of this paper, there's no math, there are no graphs. This paper is unique, and it's common for a developmental paper, but it's unique among the papers we're likely to read because all of the analysis is visual. They've photographed skeletons, and they want to show you the impacts of having these mutants, comparing a control mouse to a mutant mouse. So in this case, we're looking to start at the HOX10 mutants. Now remember, they did 2 different paralog groups. They did the HOX10 group and they did the HOX11 gene paralog group. So here we're going to start with what HOX10 does. And they note that in this triple mutant, there are no lumbar vertebrae. So here are lumbar vertebrae in the control mouse. There's nothing that looks like that in the mutant mouse. And so, for example, here's an example lumbar vertebra. And we don't see anything that looks exactly like that in shape in the triple mutant mouse. So there are no lumbar vertebrae. They also see that the lumbar vertebrae don't have rib processes. It doesn't look like those vertebrae are starting to try to grow ribs, but there are in this triple mutant, which is highlighted by that yellow bracket or parenthesis, brace. I'll get my terminology down here pretty soon. So basically, these mice look different. The mutant is different from the control mouse, and it's growing some rib processes, and it's missing a type of vertebra. So it seems like the axial formula has been screwed up in this mouse. And then here, when we look down at panel P, which is separate from that panel we were just looking at, but in the same figure, still figure 1, this is a single mutant with one wild type allele. So I believe they call this the 5 allele mutant. So there's one wild type, that uppercase A. So you can compare this animal to the control animal and look at the effects there. And it turns out that when you even have a single wild type allele of the 6, that's enough to make the phenotype of that animal much less severe than the triple mutant, where all 6 alleles are mutated. So now we can look at the other mutant that they looked at. We just talked about 10 briefly. Here's the HOX11 triple mutant compared to the same version, the same panel, the control here down below. So we can see that the phenotype of this mutant mouse is equally severe, according to the authors, to the HOX10 mutant mouse, but it has distinct phenotypes. So it has no sacral vertebrae. So what are sacral vertebrae in the wild type mouse? There's nothing that looks quite like those vertebrae in the triple mutant HOX11. So that in itself is quite strange. They also see that there are no rib processes like there were in the HOX10 mouse. So at this point, the authors have some visual information that's pretty compelling, in my opinion, that both HOX10 and HOX11 are important for axial patterning, because when you mutate them, you change the identity of the vertebrae. And then if we look for a second at figure 1Q, again, this is a separate single wild type allele mutant, where it's got 5 mutant alleles and 1 wild type. In this case, it's one of the alleles of HOX11C. And they find that it also has sacral wings. So this is, again, different from what happens in the HOX10 mutant if you go back and look at that panel in this figure. So the authors at this point feel pretty safe concluding, I think, that the HOX10 and the HOX11 phenotypes are different, which suggests that those 2 genes have different roles in development. So they change, and this is an important point, the mutations are changing the identity of vertebrae. We're getting rid of some sacral, or we're adding some lumbar, but the number of vertebral elements isn't changing. So it's not making the spinal column have more or less, more or fewer vertebrae, for example. It's these mutations are just changing the identity of those vertebrae. And here in figure 2, the authors are interested in understanding something about the pelvis and where the pelvis connects to the spinal cord. And they find that so these are the whole animal photographs taken from the side of the animal. And we can see the pelvis developing. And so the panels on the right are the dissociated pelvic bones and structures so that we can see them more clearly. And the authors simply point out that compared to the control animal, the 2 mutant animals have patterning perturbations, as they say, patterning defects. But they also point out that even though these mutant pelvises develop differently and they're differently shaped, and that's pretty obvious, I think, they still connect at the same axial position along the spinal column. So this is true both for the HOX10 triple mutant and the HOX11 triple mutant, even though it still has, again, maybe much more severe patterning defects compared to control than in the HOX10 triple mutant. They still have their pelvis connected at the same spot. So they conclude, and for this experiment and analysis, that the positioning of the pelvis and the limbs isn't under the control of HOX10 and 11. So it seems at this point like HOX10 and 11 are important in axial patterning, the identity of vertebrae, But even if they're changing the identity of vertebrae, that somehow is not affecting where the pelvis and the limbs develop along that axial axis. How's that for redundancy? And before we get into the last set of figures, it's probably useful to go over a little bit of limb anatomy terminology. So those of us that have limbs, sorry snakes, although they do have some rudimentary limbs, we can divide our limbs, both the forelimbs and the hind limbs, into 3 different developmental zones: the stylopod, the zeugopod, and the autopod. So the stylopod is essentially the upper arm or upper leg. In this case, we're looking at a chick wing. So that would be a forelimb, not hind limb. So this would be the femur in a hind limb, for example, our upper leg. And then, so that's the stylopod. The lower limb is the zeugopod. And then what's essentially the hand or the foot in a mammal, most mammals, I should say, is the wrist and then the digits, the carpals or tarsals and the digits. So a little bit of anatomy before we take a look at and discuss figure 3. So now the authors have switched from looking at these mutants, and again, we're going to be looking at both HOX10 and HOX11 compared to the control in the middle. And now we're not looking at we're looking at the triple mutants, not the ones that have a single wild type allele left. So pure triple mutants of these mice. And so again, we're looking at Alcian blue and whatever the red dye was. What was the name of the red dye that we were using? It was alizarin red and Alcian blue. So let's see what happens and see if you can find out some obvious defects in these mutants between the controls and the 2 mutants. So let's start with HOX11. HOX11, we've got there's a typical femur, short femur in the HOX10, and that's still femur. So short femur in the HOX10, no effect in the HOX11 mouse. But then look at the forelimb, right? Control, 2 bones, as normal, kind of deformed development in the HOX10 mouse, but no forelimb to speak of at all in the HOX11 knockout mouse. So they're basically missing the zeugopod. It doesn't look like in the HOX11 mutant. So that tells us essentially what the HOX11 paralog group does, or one of its roles at least, in limb development. It is going to be important in zeugopod development. Another thing that they mention, which does also speak to the development of the upper limb in the HOX10 mutant, is they mention that it doesn't contain the deltoid process, which I'm not an anatomist, I believe is this process on that bone, which is present in the HOX10 or HOX11 mutant, but not present in the HOX10 mutant. They also mentioned that when we look at the hind limbs, there are clearly hind limb developments as well, comparing the wild type to either of the 2 mutants. And interestingly, in this case, it looks like the pattern is reversed, where the HOX10 mutant doesn't develop the upper limb, where it did in the forelimb. It was shorter, but it still developed, but it doesn't develop in the hind limb. And the HOX11 mouse maybe develops a bit of a forelimb, but still not like a wild type forelimb. The other thing they point out in this example is that where a normal mouse would have a patella, a kneecap, right there, that doesn't develop in the HOX10 mutant. So basically, the HOX10 and the HOX11 paralog groups are required for patterning the stylopod for HOX10 and the zeugopod for HOX11. As is typical for many scientific publications, the last figure is often a cartoon or a diagram of what the scientists think that they've learned, a model of how their data can incorporate and either confirm or contrast with other already published literature and findings. And in this case, the authors are simply indicating where, based on the results of the mutants we just looked at, which paralog groups they think are responsible for the development, proper development of which part of the limb. So we just saw, for example, the roles of HOX10 in development of the stylopod and HOX11 for the zygopod, and some effects then less severe, more minor effects of the HOX10 group on zygopod development and so forth. And they see that these patterns are fairly similar between the forelimbs and the hind limbs, but there are some important differences. And of course, they didn't look at HOX9, 12, or 13 in this paper. So these diagrams contain the addition of data from other previous studies that, in this case, for example, suggest that HOX9 has no role in hind limb patterning, where it does have a role in stylopod development in the forelimb. And so it's maybe surprising, but it is true in developmental biology that the same genes aren't used to control the fates and development of things that you might assume are similar, like forelimbs and hind limbs. Because as you might expect, and as you know, human forelimbs and hind limbs are different. So we wouldn't expect their gene expression patterns and the genes that control their patterning to be identical. We would expect them maybe to be similar. And that is essentially what this model is representing. So it's a stylized conclusion of the paper depicting or diagramming how the author's data fit into other known data. Likewise, we can also take a look at the other component of that figure, which was the axial patterning figure. And this, I think, is really important because it points out some gene regulatory cascades down here at the bottom. So let's take this slowly and make sure that everybody's on the same page. The axial equation or formula here indicates the number of cervical, thoracic, lumbar, sacral, and caudal vertebrae, or at least a cartoon of them. And that's supposed to represent the order and number of vertebrae in this control panel. And so we should note first that, and the authors already mentioned, that the lumbar vertebrae don't grow these extensions that look like they're rib processes. And that's represented here. So the lumbar vertebrae tend not to have those rib processes. The thoracic vertebrae all have rib processes in a wild type mouse. And then we've also seen that there are those sacral wings, I think, if I remember correctly, that they call them. So that would be a wild type mouse. So what happens when we get rid of HOX10? They grow rib processes on the lumbar vertebrae. So the loss of HOX10 causing the growth of these rib processes means that when HOX10 is present, it's repressing rib development. And that's why the authors indicate that here, that they're hypothesizing that based on the results of this mutant mouse that doesn't have HOX10, that when the mouse does have HOX10, when you go from the mutant to a wild type worm, the ribs disappear. So they propose that HOX10 represses development of those thoracic ribs. But then we still have to deal with the fact that there are these sort of rib-like sacral wings that do exist in wild type mouse. So they then also have to explain, well, how did those develop in a wild type mouse if all HOX10 does is repress the production of ribs like it does here in the lumbar vertebrae? So then that's when the authors suggest, well, maybe HOX11 is important for causing the development of those processes, because when you get rid of HOX11, those disappear. So they suggest that HOX10 is necessary to repress the development of those rib processes. But in this tiny area, this relatively small area of the vertebral column, HOX11 expression is on. That transcription factor is being produced, and one of its effects then might be to turn off, just in that region, it represses the repressor. So that double negative feedback is, if HOX10 is normally repressing rib development and you repress the repression of rib development, you've activated rib development, or sacral wing process development in this case. So to conclude, there are a couple ways, major ways, that this paper is relevant to both the field of evo-devo and also to our class. One is it discusses the big question of what is the fate of duplicate genes. When you have paralogs, do they neofunctionalize, subfunctionalize, and so forth? So this paper directly addresses that big question. And as we just discussed, it also suggests the presence of regulatory networks, which we've briefly discussed so far, are important for understanding the regulation of development, because genes will repress or activate other genes, and those networks of genes cause developmental patterning and cell fate decisions. So I have a few things to point out that are, I'm going to classify them as critique. They're just things to consider. They're not all necessarily critique points. So for example, one is the authors mentioned that they had 2 to 7 skeletons per genotype. So what we should think about as, well, the paper's published, but I always read a paper as if I was a reviewer, one of the peer reviewers, before this paper is published, when the manuscript is submitted to a journal and the editor wants the expert opinions of people in the field to agree or not whether or not the paper was done properly, whether appropriate statistical tests were done, or whether there was enough replication, whether it's written well, whether it's cited all of the appropriate research. So is 2 to 7 skeletons enough for replication? That's up for you and I to decide on our own. We can definitely discuss that in class if you want. Related question then is, when you have replication, but a publication like this, especially one that's this old, and in a journal like Nature, which has a short page count for manuscripts, how did the authors choose which of those 2 to 7 skeletons to show us pictures of? Did they select truly representative ones, or did they pick the best ones from the set? That is, are we looking at the most severe phenotypes? We don't know, for example, just from what we've seen in this paper, if there might be a variation in the phenotypes of stylopod development in HOX11 triple mutants. We saw one picture of one embryo. What did the other ones look like? Did they all look the same, or was there a wide range of phenotypes? That's something we can't tell from this paper. The paper also, and this is less of a critique and more just a point for you to understand, they don't address, and I don't think they meant to, so that's why it's not a critique, but this paper, I caution you, doesn't address the big question of what types of genetic variation are important for evo-devo. So that's a big question of evo-devo. It's not something that this paper set out to address, but we still don't know from this paper whether regulatory mutations versus coding region mutations might cause axial patterning evolution. Because they didn't look at natural mutations, they caused particular types of mutations to create these mice to study. So their experimental approach wasn't amenable to answering this question, and it's a question that we shouldn't assume we can learn about from this paper. Also, and again, this is less of a critique because it's not important, because they did find really cool things when they knocked out these genes. They didn't justify why they studied these 2 genes, and that's something that's useful sometimes, most of the time, to pay attention to in the introduction of a paper. You should pay attention to, is there an intellectual justification for why they picked these genes, as opposed to, say, HOX9 or HOX12. Likewise, why did they use mice and not some other organism? So those are things that you should expect to find justified in publications, and if they're not there, you should be a little bit skeptical and ask maybe more deeply, or think about more deeply, why is it that that organism was chosen or those genes were chosen? Is there something else that the authors aren't telling us that we should know about? A few other cautions here is that the authors did collectively eliminate all of the paralogs. So that tells us the role of the group, the whole set of paralogs. But they also mentioned that the paralogs are redundant. So what's important to keep in mind, another thing to keep in mind, is that these sets of mutations are unlikely ever to exist in nature. Obviously, these are synthetic mutations. The authors created these by genetic engineering mice. So these phenotypes, the traits that we just looked at, the weirdly developed, the sacral wings, the rib processes, the weirdly developed stylopod and zeugopod in these 2 different mutants, those mice will never exist in nature. So these types of mutations, missing an entire set of paralogs all at once, that'll never happen. So these aren't the types of mutations that will ever be subjected to natural selection. So although this paper studies evo-devo and it's relevant, this doesn't actually tell us about how evolution occurs. This is all synthetic work. It's not based on naturally occurring mutations. So I suggest that it's probably still true that evo-devo works through minor changes at a time. And you might imagine that if you wanted to have a mouse that had longer tibia, for example, or tibia and fibula, or a longer femur or a shorter femur, because it's adaptive, let's say it's a ground-dwelling animal, it buries tunnels in the ground, and so it needs to have shorter limbs. You might expect that small mutations to how these Hox genes are expressed, or where they're expressed, and how much they're expressed, how much protein is produced, could make those sorts of changes by slowly, over time, making minor adjustments by mutations that have small effects. After generation, maybe you might eventually create a mouse that has a short femur like we saw in one of the mutants. And I always like to end an interpretation of a paper by thinking about what didn't the authors do and what might they be interested in doing next. And so in this case, I have this big question of what's going to happen if somebody ever mutates other Hox paralog groups. Let's look at Hox 12 next, maybe, and see what happens. Now, for this first presentation, I do want to make sure that I go over, again, the rubric, the expectations for these sorts of presentations. And this is the rubric from the syllabus. And I want to briefly mention why I think I covered most, at least, of these rubric points. So enhanced with rich media. I added the figures, and you also noticed that I added some additional figures, like the one of the chick wing development, to introduce some vocabulary. So that's an example of rich media, embedding videos that are relevant and so forth. We did talk about the research motivation, that nobody had ever been able to knock out all of the Hox 10 and 11 genes, so we didn't know what they did. That's also the novelty, that this was a technique-based approach, that they were able to get rid of all of the paralog, an entire set of paralogs all at once. And discuss the relevance of the paper to evo-devo, that is understanding how the axial pattern is created. It's a fundamental question in developmental biology. And although there was no hypothesis, often evo-devo studies are discovery-based. So instead of a hypothesis, I don't remember exactly what I called it. I think I called it an objective, maybe. They had an objective, so they didn't have a prediction of what they were going to find out when they knocked out the Hox 10 paralog group and Hox 11 paralog group. That was the objective of the study. So that's equally reasonable compared to a hypothesis. We talked about the experimental design, things like how they generated the mutants by inserting a marker gene, for example. The research methods, that might be, for example, talking about the staining of the mice that I did. That they used alizarin red and Alcian blue, what those stained, that these were embryonic mice. And then we also discussed, in this case, there weren't many analyses. The analyses were all visual and comparative, but the types of data. So a description of the different bones that they were looking at and the individual comparisons. One thing that you're going to hear a lot from me about in any class I teach is when you analyze any sort of figure or table, anything graphical or anything presenting data in a manuscript, the authors are presenting all of the data or much of the data they collected, but there's usually one or two contrasts or comparisons they really want you to make. And that's what's going to be in the written form of the manuscript usually. So when you read the manuscript, they will point out specific aspects of a data table or figures. Like, for example, when they mentioned that the patella developed in one mouse, but not another mouse. It's a very minor thing, but they wanted to point it out because it's developmentally important. So those sorts of data comparisons are things that I expect you will point out. If there's a ton of data, don't feel I didn't present all of the panels. There were some panels I didn't even talk about. So don't feel like you need to focus on every single panel or every single element of a data table. Pick the ones that are important. We talked briefly about drawbacks to the research and limitations of the interpretations of their data in the conclusions that I just gave. And I did mention things like this involves questions about the fate of duplicate genes and about gene regulatory networks. Those are kind of big topics that will become threads as we read additional papers in the class. So I hope that's a useful walkthrough for you in terms of seeing what I expect and how I'm sort of evaluating the rubric that I've laid out. I don't expect your presentation to look exactly like mine. You don't have to have the same headings, titles. Another thing I should mention briefly, you might notice that I put some of the figures out of order. And I discussed some of them not in the same order that the authors mentioned them in the paper. That's also totally fine. It makes more sense to present the data in a logical order than necessarily in the order that the authors provided. So to conclude this paper, here are a couple things I'd like you to think about. Maybe we'll discuss them in class next time for those of you that show up. Do you think after reading this paper that there is indeed a Hawkes code for axial anterior-posterior patterning? Why do you think the authors chose to work in mice instead of another organism like chicks or other vertebrates, salamanders, for example, who knows? And why do you think this paper was able to be published in one of the most prestigious scientific journals, Nature?